Whether you are unicellular or multicellular, you need to undergo some type of cell division to continue life. Unicellular organisms like bacteria need to divide to reproduce, whereas multicellular organisms such as yourself need to grow from one cell to 37 trillion cells. But how do you do this without making some major mistake along the way that just completely changes your course of evolution? Well, before a cell can divide, all its DNA needs to be replicated. Then the two copies of DNA need to be separated into two separate nuclei so that each daughter cell has a complete genome. For example, human somatic cells have 46 chromosomes each that are made up of two sets of 23, one set inherited from each parent. Now this part is mitosis. Finally, the cytoplasm needs to divide to create two separate cells. This is cytokinesis. Now let's unpack each of these. Interphase is organized into three parts, G1, S, and G2. Firstly, the cell grows by producing proteins and organelles like mitochondria and endoplasmic reticulum. This is G1. Secondly, the DNA duplicates itself through DNA replication. One chromosome becomes two, and they are identical, so they are both called sister chromatids. And these two sister chromatids are joined together at the centromere. I'm only showing it condensed like this, so you can actually see that it's gone from 1 to 2, but it's actually loosely packed in a form called chromatin. Thirdly, DNA is checked for mutations in the G2 phase. Genes like DNA repair genes, proto-oncogenes, and tumour suppressor genes, they would be checking DNA for mutations, and if they pick up on any mutations, they would actually slow down cell growth to either fix the mutation or to completely start all over again. Now it's time for this DNA to completely separate into two nuclei, and that is through mitosis. Okay, firstly, in prophase, chromatin condenses into visible duplicated chromosomes. The centrosomes begin moving to opposite ends with fibers forming between them, and the nuclear membrane breaks down. Secondly, in metaphase, spindle fibers attach to the centromere, and these fibers arrange the chromosomes along the metaphase plate, which is the center of the cell. Now, thirdly, in anaphase, the sister chromatids are pulled apart towards opposite poles, so they become independent chromosomes. Lastly, in telophase, the nuclear membrane reforms, the nuclei moves to opposite poles, and the cell starts to pinch inwards at the center. Now, to finalize cell division, the cytoplasm needs to completely pinch to separate the nuclei to produce two genetically identical daughter cells. Now, I'm going to show you some pictures of a plant cell undergoing mitosis, and you're going to tell me what stage it's in. This image over here, you can see that the chromosomes are lined in the middle, so it is going to be metaphase. What about this one here? Because you can't really see the actual, condensed form of the chromosome, and it just kind of looks like a lot of black space, this is because they haven't condensed yet, so they're still in interphase. What about this one? Students actually always think this one is either between anaphase or telophase, but it's actually telophase because you can see a nuclear membrane around it, and you can also see that the cells started to pinch. So you can see that bit of a curved cell membrane there. Now, if a question ever asks you about the purpose of mitosis and why a living organism would need to undergo this type of cell division, you need to remember RAGE. R for repairing damaged tissue. So if you fall and you scrape your leg, those skin cells need to reform that barrier by producing more skin cells. A for asexual reproduction. For example, bacteria undergo something very, very similar to mitosis, which is just binary fission. G for genetic stability. Mitosis makes sure that the replicated DNA equally distributes into two daughter cells, making sure that each cell has a complete genome. So make sure there isn't too much DNA in there or too little DNA. And E for embryonic development. This is where, after fertilization, the first diploid cell, that is called the zygote, needs to continue to divide via mitosis in order to go through all the stages, including morula, blastocyst, gastrula, and then fully developed fetus. Anyway guys, so this is the end of the video, so I hope you really enjoyed and I hope you learned something.